Okay, recording and everything good. Okay, so let me show you where it is. You whip down to um, I isn't. Okay, so if you go to files, unit 3, slides, it is this one here, enzyme classification.
Needs that much more time.
1,000 words on the essay. Well, let me talk about that. Next week, if you have your project done, you may bring it in on Monday or Wednesday. Excuse me. Being able to be glad to grade at least the poster part. And I have to read the essay, of course. Or any time between now and December 11th, which is the day of our final. Okay. So basically, I'm looking for a 3-panel poster. And with examples, if you go on to if you go to Canvas project under files, you'll see some examples. Think of the poster as multimedia for your paper. And then on the paper, just a standard, what is it, MLA or whatever that standard the English department uses. That's real standard stuff, nothing special at all. Yeah, this assignment is due December 11th or sooner. So it's not due next week. If you've got it done, you can bring it in next week and be one less thing you have to worry about. Okay? That's what I meant by that. That makes sense? I'll post a rubric, which is very simple. Let me see if this. Yes, you can bring it Wednesday also. I'll publish a more detailed rubric than this. This is very generic. And yes, you can bring it in this Wednesday. That's fine too. You can also take 4 images of your poster, 1 for each panel plus the overall picture, and then send and post that under assignments, which I'm going to put there right now. This is what it looks like to create an assignment. Okay, so if you'd like to do it via upload, it's available. Otherwise, just bring it in and we can chat about it. There's no presentation, okay? A lot of you have been asking me, do I have to present to the class? No, all we're gonna do is you're gonna show me your poster and I'm gonna ask you questions about it a little bit. If there's some clarification, just, excuse me, a very, very low-key talk, okay? All right, so any further questions? I'll try to get that done tonight on posting the detailed rubric. I thought there was on the sunscreen lab. Let me just check real fast. Let's see, that lab was fixed. Huh.
Yeah, I would say that, like, yeah, they used to get the 2 Korean tacos. I'm like, what is this? I sometimes I get my coworker and I was all right before we're ready. And then
Okay, so let's go ahead and get started on today's content.
yeah, I'm pretty sure it's just it should just go by K. Okay, so that's K. Okay, thank you. Okay, I appreciate it. Okay, thank you. Bye-bye.
Okay, everyone, please mute your Zoom.
Yes. Oh, you're so sweet.
Melissa, you're not muted.
Thank you. Okay. This series of slides comes from the slide collection based on your book. So this is section 19.3 out of your book. And they did a much better example than the other slides I was using. So that's why I'm incorporating this set in conjunction with the others. Okay. There are 7 main classes, but 6 of them are the ones that involve reactions. So the 7th one, we're not going to worry about. Okay. They're based upon the reaction. Remember, a reaction in biochemistry is you have substrate, plus the enzyme gives a substrate, enzyme complex, which is also called activated complex. And that yields yields the enzyme back. So the enzyme is unchanged. If it's changed, it's not an enzyme. It has to be regenerated once it's done. So basically it pulls in the substrate, it does its reaction, spits out a product, and then it's the enzyme again. Okay? All right. So this is what they're talking about, the reaction. Okay? All enzyme reactions are in this form. Just like in 305, we talk about reactants and products. In biochemistry, we talk about substrates in products. same kind of deal. In 305, there was the activated complex with the reactants bonds being broken, forming an activated complex, and then from there forming the product or back to the reactant side. Okay, same with enzymes. Enzymes are the catalyst. That means they speed up the reaction by lowering the activation energy. Okay. There are two parts usually, I'm going to say usually, to naming an enzyme. There's two parts. One lists the substrate, and the second part lists the enzyme class. So the substrate is what are we working on? For example, if we're working on ethanol, and we're oxidizing ethanol. So remember, ethanol looks like that. It's a primary alcohol, easily oxidized to an aldehyde. And that's easily oxidized to the acid. So in our liver, when we oxidize ethanol, it takes two enzymes to do that, to get it over to the acetic acid. It takes a dehydrogenase, which I'm going to talk about in a second, and an aldehyde dehydrogenase. And this is the culprit that lacks in some people. They have the alcohol dehydrogenase, it's the aldehyde dehydrogenase that they lack genetically. Okay, so this page is out of your book. It lists the 6 classes of enzymes, and it also lists the subcategories from each enzyme. For example, here's a dehydrogenase, which is mentioned here. The removal of 2 atoms to form a double bond. Okay. Now, one thing I want to mention also is this other chart. So when you look at an oxidation reduction, this is what's going on. What happens is these electrons go there. So I'm going to talk about that in a second, though, but this is it more succinctly. I'm going to be using the A and B's as well in the second here, but these are all summarized for you. Okay, so let's look at oxidation reduction reactions. Those enzymes generally are called oxidoreductases, like oxidation reduction, oxidoreductase. Okay, in biochemistry, we don't usually talk about the transfer of electrons as we do in inorganic chemistry. We talk about the transfer of hydrogen and oxygen instead of. Now, because it's an oxidation reduction reaction, if something's being oxidized, like that ethanol going to an aldehyde, if this is being oxidized, we have to have something being reduced at the very same time. That's what they mean by a coenzyme. That's going to be what's either oxidized or reduced if your substrate is being oxidized or reduced. So in this case, the alcohol is being oxidized. See, we're losing a hydrogen off of that oxygen there. So what's going to be reduced? There are 4 reduction coenzymes that are frequently used. I'm going to talk about these next time we talk about carbohydrate metabolism. And the most common one is this guy and that guy. And that's a positive charge. So what happens when that's a D also. When NADH plus goes to NADH, what happens is we gain a hydrogen. Up here, we lost the hydrogen. So what we do on one, we do the opposite on the other. Yes, the recording is on. Okay. There are 3 subclasses of oxidoreductases. There's a straight oxidation, straight reduction, and then a more specific one on focusing in on hydrogen atoms. So this is the reaction I was just talking about. We have ethanol, and we have ethanol, aldehyde. Okay, so that's being oxidized. This is being reduced. This is going to lose a hydrogen. Gain a hydrogen. So in this case, that's the coenzyme. It works. It goes from an oxidized state to reduced state. Okay. Transfer bases. Transfer. Key in the word transfer. And there are 2 subclasses. Transaminase. So that transfers an amino group between the substrate and the product. And a kinase transfers a phosphate group to the product. Okay. So if we had something like what enzyme would transfer a amino group to here. That would be a transaminase. It transfers an amino group. Okay, so step number one is to figure out, looking at the substrate and product, what's being done, and that will determine enzyme classification. Kinase transfers a phosphate group, and that's classic when we're talking about ATP and ADP. For example, the third step of glycolysis is this here, where we have a phosphorylated fructose, and we become, and based on the kinase, we've added another phosphate group on the carbon number one. And then to do that, where does it get the phosphate group from? From an ATP. So the ATP becomes an ADP. That phosphate group is transferred up there. We're going to see this again when we talk about glycolysis. There are three main steps to carbohydrate metabolism. There's glycolysis, Krebs cycle, and electron transport or oxidative phosphorylation. Those are the three main groups we're going to talk about in intimate detail. Okay, so those are the two kinds of transferases, the aminases and the kinases. Okay. Here we're talking about hydrolysis. Now remember, hydrolysis breaks apart esters and amides. And this is, the esters are important in terms of fats, amides, proteins, and then we have esters in terms of RNA. The nucleotides are tied together via phosphate esters, the deoxy and ribose sugars. Okay, so lipase is the enzyme that breaks down fats. It saponifies fats down to glycerol and fatty acids. Proteases break up proteins. They hydrolyze the peptide bonds, which is an amide bond, to amino acids. And then nucleases breaks up the DNA and RNA nucleic acids, which is esters. Actually, phosphoric esters. Okay, and the ribose the ribose and deoxyribose sugars through esters, phosphoric acid esters, tie those bases together. So we get into mRNA, tRNA, and DNA. This all involves nucleases. Okay, so for example, if we have a polypeptide here, And this is the acid end, or the C-terminal end. We put this in the presence of a protease in water. We're going to end up cleaving let's see. Leaving that amide bond right there, giving us a free amino acid and a shortened peptide by one amino acid. And then we had a triglyceride with a lipase. That lipase then would cleave that ester bond right there, so we end up with three either three fatty acids or three fatty acid salts, depending on whether this was an acidic environment or an alkaline environment. Plus glycerol. And you'd have something very similar and that's what ribose looks like. This is going to be part of our lab today. And when that would team up with in this position, a esters to tie the strings along. And over here, you're going to have the base. So those are the three classes. This would be a nuclease would break this ribose backbone up into individual base units. Isomerases are probably the simplest ones. Basically, isomerase just changes the position of the functional group within the molecule. For example, here we have the carbonyl in the middle. Here we have the carbonyl on the end. To move those around, it's a complicated reaction from a mechanism standpoint, but it's easy for an enzyme because they're already set up to do that. So the isomerase here, the triose phosphates are going to be moved around. Liases, there are four subclasses. A D-carboxylase, catalyzes the removal of a CO2 group. Deaminase, removal of an amino group. Dehydratase, removal of water. And then a hydrase is the addition of water. So in this case here, we have a double bond and we're adding water. This is just like unit 1 reactions, only this time it's with an enzyme. So you have fumarate, which is a double acid, diprotic acid, with an alkene in the middle, this double bond. And as you remember from group unit 1, when we add water across the double bond, we create an alcohol. Excuse me. So in this case, it forms, because it's in the middle of the backbone, it's going to form secondary alcohol. And then, of course, we could get a malate as well, which would be hydroxyls on the same side. But with a reaction catalyzed by an enzyme, we don't get that because it's done via an enzyme. If it was in a test tube, we would get a mixture of the two. So enzymes are horrendously specific. Let's see, ligases. All right, now they catalyze the formation between two substrates, okay? Sometimes it's forming a phosphorylation, for example. For example, down here. In this case, it's a carboxylase. Basically, we're adding a carboxyl group to the substrate somewhere, okay? And in this case, we're adding it to this tail end methyl group right there. That's being added to that via this, and this is pyruvate, so it's pyruvate carboxylase. And then if we get the required energy from the ATP, it's got to lose the phosphate. In this case, the phosphate is going to just a generic phosphoric acid. Okay, so sometimes ATP to ADP, we actually put a phosphate group on something, or when we lose that third phosphate group, all we're getting is energy. Okay, so reason number 1 is we're like the 6 fructose 6 phosphate, and then we're moving it over to a 1,6-bis phosphate. Okay, number 2 is we're just needing energy. And then when we go from ATP to ADP, we're releasing a phosphoric acid from the ATP. So when we break that bond, it releases energy. Or we break the bond and transfer that phosphate group to another molecule like we did up there. This guy right here. We have fructose with 1 phosphate group, fructose with two phosphate groups. The phosphate comes from the ATP. Down here, the ATP is just supplying energy and is not interacting with the substrate. Okay, so those are the 6 classes and subclasses of enzymes. You know, 6 main classes with subgroups. Okay, so we have a reaction here. We've got this 3-carbon amino acid. And we have a what would this be? This is going to be a dual acid with a carbonyl group in the middle. And what we're going to do is, what change occurs in the product? Well, that amino group is transferred to that carbon there. So when we transfer a group, the enzyme transferase is the one that does it. So we have to look at what's going on between the substrate and product. Okay, this has no group. But it has an extra carbonyl added. So in other words, the NH2 and the C double bonded O swap places. So if you look in your book, on page 593, there's some problems. I'm gonna do a quickie picture of this page and move it up. For 1 second, I need to transfer that over. Oh, it's not. Okay. Okay, so if we look at the names of these, remember the names come in two parts. There is a substrate, and we have the function. In this case, it's alcohol dehydrogenase. Dehydrogenase is a redox, and that's oxidizing. Dehydrogenase means you're removing a hydrogen from an OH group. So we're taking an alcohol to an aldehyde.
You're not sharing your screen.
Oh. Thank you for telling me that. Can you all see that?
Yeah.
Okay. All right, so let's start over. Did you have any idea what was going on? Okay, so when you see the name of an enzyme, usually it's in two parts. Here's the substrate. And here's the reaction. Okay, so the substrate in this case is an alcohol. And dehydrogenase removes one hydrogen. So when you remove a hydrogen, that's oxidation. And when you remove one alcohol, when you remove one hydrogen from an alcohol during oxidation, you produce an aldehyde. And we can do further reactions if we had an aldehyde dehydrogenase, which removes one hydrogen from the aldehyde, forming an acid. And as I said numerous times in this lecture and last week as well, that this is how your liver detoxifies alcohol when you're drinking. So if you lack either one of these enzymes in your liver from a genetic reason or cirrhosis, the alcohol is going to do damage in your system. If the alcohol can pass the blood-brain barrier, it can get into your brain, and that's why you get drunk. But it's also a poison. So keep that in mind. Just make sure your liver is healthy. Okay. Does that make sense on breaking this enzyme down? Okay. So here's our substrate. And what does a synthesase do? Well, let's go up here. To our original chart. Okay. So this is adding usually water across a double bond. Okay, so these remove, this adds. We're going down to this problem. Remember, the tRNA is made up of phosphoric acid esters with the sugar ribose. And what we're going to be doing is adding one of the double bonds or all the double bonds. And we're just going to break up that tRNA. Okay. Substrate. Transaminase. Okay. What does, I'm going to take this chart and put it to the right of this. So we want to keep going up here. So here we have transaminase, and that transfers an amine group between substrates. So it's going to take the amine group off of aspartic acid, amino acid, and transfer it to something else. So transaminase needs its source of the amino group. Here it's getting it from the amino acid. Okay, so we have here's a 6-membered monosaccharide with a phosphoric group on it. And that isomerase is going to move the phosphoric group to another carbon location. So if you look at the isomerase over here, we're going to just move things around. Okay. And what class of enzymes do the hexokinase belong? Okay. Remember over here, a kinase transfer over a phosphate group. So if you remember, we had fructose in position 6, and then we used a kinase to add a phosphate group onto position 1. So we had phosphate groups on positions 1 and 6. Okay. Um, okay. Hmm. This is an interesting problem. There is no lyase catalyzed reaction on page 592. Okay. All right, which following reaction can be catalyzed? Okay, first of all, let's look at this here. We have substrate. And we have product. What's the difference? This is gone. That's the difference. So this is going to be numeric acid. Okay, so the decarboxylase removes carbonyl group. So if we look over here, replacement of a carbonyl group by a hydrogen. Okay, so that's what's going on here. That is removed. Okay, part B. Okay, we have, this is a non-standard amino acid. And what's going on here? Well, amino group. I guess my dryer says my clothes are done. The amine group is gone. Well, what removes the amine group? Well, let's see here. Transaminases removes an amino group because it could transfer it over to another substrate. What else removes amines? Well, let's see here. That looks like the only one that will take off an amine group. So this would be, so you have to have two substrates here. And that would be transferred to that. And that would be trans. Bottom line in this is you have to look at what's going on with the molecule. Um, um, and then you go over here and see which one correlates to it. Now, as we go through the metabolism of carbohydrates, we are going to see lots of examples of this. In the beginning here, you're saying, well, aren't there many examples? You know, it's just kind of hard. Okay, well, just hang in there. And when we get into actual carbohydrate metabolism, you'll see how each one of these is used. And that's where the important part is. You'll see what's going on. For example, when glucose first goes into being metabolized, what happens? Well, first step is you add a phosphate group, carbon number 6. So it becomes glucose 6 phosphate. Anyway, so there's a number of steps, and then that enzyme would be a kinase. It's adding a phosphoric acid group. Okay, so that's where I want to leave this today. I've introduced you to 6 categories, and then when we get into carbohydrate catabolism, actually it's not metabolism, it's catabolism. We are going to then look at these enzyme classes once again, so we'll have more repetition of that. Okay? All right. How are you guys doing? Okay, on your lab today, your lab write-up today and Wednesday, there is a flow chart sort of in the beginning. It's on the second or third page. That's a really important flow chart because you're going to be using that. You're going to be going through each step of the flow chart with reactions associated with each step. Then you're going to have an unknown, and you're going to take that unknown through the flow chart. You did a flow chart before looking at what was trying to think what lab that was, but you had a flow chart. Same idea anyway. So you start with something, you're going to do one reaction on it. You're going to get a yay or nay, yes or no. If it's a yes, you're going to do something else on it. If it's no, you do something else on it. And you're going to continue down, you end up identifying your unknown. So make sure you have that flow chart in your write-up. Okay. All right. Are there any more questions about the project? Any more questions about well, I hope you all took advantage of correcting your test 2 and submitting those. Now, the assignment said 1 point, and the reason why I assigned that is 1 point. You can't assign 0 points because it's going to change your test 2 grade. You're not going to get a grade on your correction assignment. You're going to get a grade change on test 2. And that's a 50% rebate, basically. So you can get half the points back, assuming you get the problems correct. Okay? Well, if I knew who iPhone was, maybe.
Oh, it's under iPhone. I'm sorry, Professor. It's Imelda.
Do you have lab today?
Wednesday.
Wednesday. So you did not get your lab or your test back?
No, I wasn't in lab last week, so maybe that's why.
Yeah, because I gave them on in lab. In fact, I gave Wednesday's lab or test back the previous week. Anyway, I have them with me, so. Did you see your grade?
No, I emailed you about it too.
I didn't.
So I'll resend you the email.
Are you one of the two that come in from the mountains?
Yeah.
Yeah, I graded both your tests. And I'm,
Okay, it's just getting it back then.
yeah, go on to Canvas and make sure your grade's there. You both did pretty well. Anyway, if the grade isn't there, let me know, because I have your test, so.
Okay, will do.
Okay, that's it. I'm going to end this. The next chapter we're getting into, by the way, is going to be lightly hitting chapter 20 and heavy duty 21. Excuse me, 22. Carbohydrate metabolism. Okay. I will see Monday folks today in lab in a little while.